Welcome, and thank you for standing by. At this time, all participants are in a listen-only mode until the question and answer session of today's call. At that time, if you would like to ask a question, you may press star 1. Today's conference is being recorded. If you have any objections, you may disconnect at this time. I would now like to turn the meeting over to Monica Witt. You may begin.
Good afternoon. I'm Monica Witt from NASA's Office of Communications. Thank you for joining us today to talk with experts about science investigations launching on SpaceX's 24th commercial resupply mission for the agency to the International Space Station. SpaceX is targeting 5:06 a.m. Eastern Standard Time on Tuesday, December 21st for the launch of its Dragon spacecraft on a Falcon 9 rocket from Launch Complex 39A at NASA's Kennedy Space Center in Florida. Here to talk with us about some of the research launching aboard the SpaceX Dragon are Dr. Robert Dempsey, Acting Deputy Chief Scientist for the International Space Station Program, who will discuss the breadth of research being done aboard the orbiting laboratory. Dr. Michael Becker, Project Manager, German Space Agency at DLR, the German Aerospace Center, and Principal Investigator for the BioPrint First Aid Investigation. Dr. Brian Crucian, Principal Investigator for the Host Pathogen Investigation. Alicia Carey, Lead Program Manager for the Turbine Superalloy Casting Module Technology Demonstration. Dr. Imara Pereira, Research Professor at North Carolina State University and Principal Investigator. And Dave Reed, Operations Director for the MVP Plant 01 Investigation. Nico Hansen and Teo Nelson, representatives from 2 Student Payload Opportunity with Citizen Science Payloads, or SPOCs. Dr. Paul Reichert, Principal Investigator for cases PCG20. And Dr. Mark Civic, a Senior Director and Research Fellow at Procter & Gamble and co-investigator of the PNG Telescience Investigation of Detergent Experiment, or PG-TIDE investigation. Our speakers will provide a brief overview of their research, and we will open the lines for a few questions after each speaker. If we have time at the end, we can take additional questions. Your phones are on mute now. To get into the question queue, you can press star 1 on your phone at any time. We will call on you, and the operator will open your mic so that you can ask your question. We ask that you please stick to one question, and if we have time, we'll allow reporters to answer a second question. About 1 hour after we conclude, you can listen to a replay of this teleconference by dialing toll-free 866-360-7721. One more time, that's 866-360-7721. Or you can visit NASA's video YouTube channel. To start, we will hear from Dr. Robert Dempsey, Acting Deputy Chief Scientist for the International Space Station Program at NASA's Johnson Space Center in Houston. Over to you, Dr. Dempsey.
Thank you very much, Monica. I am very excited to talk to you about some of the research occurring on the ISS, and I really wish we could talk about all of the amazing studies that are occurring, but today we'll talk about a few that are very notable. But a couple general words before we get into that. For over 21 years straight now, humans have lived and conducted research on the International Space Station. It might sound odd to be talking about groundbreaking research in low Earth orbit, but that indeed is what has been happening on the ISS over these years. Now, many of you may have heard news stories about research to keep astronauts healthy while in space, such as studies to look at bone density or muscle mass. But the research portfolio on the ISS is so much more, with more than 3,200 research studies performed to date from over 100 countries. And you'll hear some exciting ones today as well. Now, personally, as someone who's been heavily involved in these 21 years, I resonate with the diverse research portfolio on the ISS. For example, as an astrophysicist, I'm excited by the alpha magnetic spectrometer probing the origin of the universe and the Japanese monitor of all-sky X-ray telescope that studies the depths of stars. But working as an engineer, I also see the power of the Advanced Combustion via Microgravity experiment, which will build better physical models, enabling us to improve car engines and rockets alike. As a NASA operator who has spent many hours in mission control, I am thrilled by the technology demonstrations that range from the spacesuit evaporation rejection flight experiment, enabling advanced spacesuits that will be used to explore the Moon and Mars, to developing 3D printing technologies that will allow astronauts to print spare parts or treat medical emergencies far from home. Finally, as a former instructor, I know that education is our future, and here too the program inspires girls and boys in programs like Zero Robotics, where students develop software code to operate flying robots on board the station, a little bit like out of Star Wars. Now, we currently have 7 astronauts on the ISS representing the United States, Russia, Japan, and the European Space Agency, including 2, Mark Van de Heij and Pyotr Dubrov, who are a little more than halfway through their nearly year-long mission helping to pave the way for going to Mars. We even have 2 paid tourists, which is just one part of the commercialization that the ISS is enabling in low Earth orbit, along with private companies testing operations or manufacturing in space. Many hands mean a lot of research will be performed with the aid of the equipment and supplies SpaceX-24 is carrying. This flight brings up dozens of investigations that will join the complement of hundreds of experiments to be conducted during the Expedition 66, representing nearly 500 hours of research. On ascent, SpaceX-24 will carry up about 1,950 kilograms in the pressurized capsule, and in the so-called Dragon trunk, it'll carry about 908 kilograms in various payloads that will be attached to the outside of the ISS. As the only vehicle that currently is able to return cargo to the ground, the 1,936 kilograms in samples and equipment returned next month are vital to complete the research or prepare for future missions. This mission also stages equipment that will be used for the first private astronaut mission to the ISS known as Axiom-1, which is scheduled in February. Now, to keep up with the space station science, you can also visit us at www.nasa.gov/ISS-science or follow us at ISS_research on Twitter. Now let's focus on a few specific studies, and thank you again for being here. Back to you, Monica.
Thank you, Dr. Dempsey. Now we will hear from Dr. Michael Becker, Project Manager at the German Aerospace Agency, who will discuss the BioPrint first aid investigation. Dr. Becker is a biologist and received a PhD at University of Cologne, Germany, in biotechnology and biochemistry. He's worked for the German Space Agency at the Department of Research and Exploration since 2015, supporting and managing microgravity-related life science research projects. Dr. Becker?
Thank you very much, Monica, for your kind introduction. I'm happy and excited to give you an overview of the experiment BioPrint First Aid. The BioPrint First Aid Handheld Bio Printer is at first a technology demonstration project to verify a handheld bioprinter for applications on Earth and also in space, of course. You might know that bioprinting is a kind of additive manufacturing using biological and living cells and also biological molecules. And the process of printing biological structures can also be used to print personalized tissue patches, for example, for treating superficial skin wounds. And therefore, the BioPrint First Aid handheld has been developed. The bioprinter is a robust and mechanically hand-operated device, and the idea is to use patients' own skin cells combined with a bio-ink to create a tissue-like patch that covers a skin wound and then accelerates the healing process. And on the ISS, this technology demonstration experiment will not use real human cells, but fluorescent microparticles that simulate the skin cells. And the bio-ink with a cross-linker and these microparticles, they form a plaster-like wound covering during this printing process. And the patch is applied on a foil that is on the leg of the astronaut, and this is in the case, this is ESA astronaut Matthias Maurer. And as you can imagine, microgravity might change the printing properties since we don't have sedimentation and other gravity-dependent processes. And the stability of this printed patch and the distribution of these microparticles that replace the skin cells in this case, and the potentially gravity-dependent cross-linking processes of the bioink, they will be analyzed afterwards. So also different printing nozzles and also bioink compositions will be compared to investigate the printing process and its properties in a microgravity environment. In addition to that, this approach is also tested with human cells on ground, and the printed samples from space and Earth will be compared and further studied by scientists at the Technical University of Dresden in Germany. The objective of this project is not only to test the print technology, but also to assess its applicability under space conditions for future space missions, and of course also applications on Earth. So such a personalized bioprinting technology could have great benefits. This includes, for example, an emergency wound treatment and improved patient care, for example, for immobile patients. But if you think of space, it might also help to overcome altered wound healing in space and improve the treatment of injuries as maybe as a part of personalized medicine and also for an astronaut-specific wound treatment. So summarized, Bioprint First Aid provides a research opportunity for the optimization of the bioprinting technology, but also its design, materials, and also its printing process. And basically it allows us to assess its relevance for to future space and exploration missions, and of course the use on Earth. Back to you, Monica.
Thank you, Dr. Becker. We will now go to reporters for questions for Dr. Becker. Please press *1 on your phone to be entered into the queue. And then please identify to whom you are addressing your question.
Our first question comes from Russell Pounds. Your line is open.
Good afternoon. This is Russell Pounds. I'm with Pacific Rim Media. I've got a question for Dr. Dempsey. I was curious, it was great hearing the review of all the science going out, and I was wondering, is there any particular point that you make when you're discussing these things with audiences or the public, community meetings, etc., when the eyes light up for the audience that you're speaking to about what's happening on station? Thank you.
That is a really good question, and I think that the main point that I like to make, because people's eyes will light up at different things, but I don't think they realize all the different research studies that are going on. I mean, we've got the Cold Atom Lab that's in the process and will be enabled further by SpaceX 24 to actually get what's called a Bose-Einstein condensate near absolute zero, where you get particles that are nearly at absolute zero, have no energy, and kind of collapse on themselves. Even if you don't understand what that physics means, that we're doing such fundamental physics on the space station, and we're doing biological sciences, and we're printing potentially, future Band-Aids for going to Mars or something. It's just astonishing all the different things that we are doing. And I think that's what a lot of people find very surprising and interesting. Back to you, Monica.
Thank you. Thanks, Dr. Dempsey. I have a question for you, Dr. Becker. I'm curious, what will the next step be after a successful demonstration?
Yeah, the next step will be, of course, first the analysis of the printed samples in space when they come back to the labs on ground. So these printed samples will be compared with the printed samples on ground, and depending on the results, of course, we plan an optimization of this printing process. We will further develop the printer in such a way that we will be able to print patches with living skin cells. Right now, we have substituted or replaced these living cells with microparticles, plastic particles. But of course, we would like to improve it in such a way that we will have living skin cells within the bioink. And the next crucial step would be to optimize the setup and the bioprinting process to print a band-aid with living skin cells on the ISS, to investigate it on the ISS. Of course, the overall goal would be to see such a bioprinter as part of a medical equipment in exploration missions, for example, like when we think of Moon and Mars.
Thank you. Carrying on with the subject of human health, we'll hear from Dr. Brian Crucian. Dr. Crucian will discuss host pathogen, an investigation that will examine the synergy between reduced immunity in astronauts and increased bacterial virulence in microgravity. Go ahead, Dr. Crucian.
Good afternoon, and thank you for the kind invitation, and I appreciate everybody's interest in this. So this is a biomedical research study. Within NASA, we have a human research program that is designed to characterize health risks for deep space missions and to mitigate them via countermeasure. And this study targets one of them, the risk for infectious diseases during deep space missions. My partner in this study is Dr. Mark Ott, the microbiologist at JSC, and I'm an immunologist. And so the background, a couple of decades of work have characterized that there is some immune compromise or some immune dysregulation when people go to space. And the reason for that is the combination of stress and isolation and circadian misalignment. And it alters their immune system, and we can measure that. The various immune cells, say T cells, NK cells, don't work as well. And we know this is a real phenomenon because we also see the reactivation of latent viruses in astronauts during spaceflight. This doesn't mean that they're ill, but they reactivate these viruses, and we can measure it. It confirms the immune problem. We also know there's some degree of individual health events during ISS missions, but that's confidential medical data between crew members and their flight surgeons. But it's not zero, although we don't have a profound medical problem during orbital space flight. Of course, the flight surgeons do a tremendous job of caring for the crew members. One example is a recent case of dermatitis in an ISS astronaut that was confirmed to be associated with the reactivation of these latent viruses. But overall, the astronauts are doing great on orbit. We're really worried about deep space missions when we increase all of these stressors. Radiation will be increased, distance from Earth will be increased, et cetera, et cetera. Much smaller vehicle. And so the study is to characterize what happens when you combine a dysregulated immune system with microbes that have been grown in microgravity where their virulence is altered. And this is some of Dr. Ott and Dr. Cheryl Nickerson's work. They found that if you culture bacteria in microgravity, their virulence changes. And that's really interesting. And we're worried about, say, the increased virulence in bacteria and diminished immunity synergizing to really affect the infectious disease risk for deep space missions. And so this specific study will try and characterize it. This is a fairly uninvestigated aspect of biology or immunology for spaceflight. And so what we will do is collect blood samples from astronauts before, during, and after the mission. They'll be returned ambient, room temperature, living blood immune cells will come down in the vehicle to our hands, our team, and we'll get the immune cells from the astronauts, and we'll culture them, co-culture them together with one of 5 bacteria that we've identified for use in this study, representative of what the most important medical pathogens might be during a deep space mission, representative bacteria. Now, the magic here is that we won't just culture the astronaut cells with the bacteria and see what happens. We're going to have normal bacteria and microgravity-altered bacteria. So we have these wonderful machines called bioreactors or clinostats, and on Earth in our laboratory, we basically rotate these cultures and simulate microgravity. So we can get normal bacteria, and on Earth we can get the virulence altered bacteria. And so pre-flight, in-flight, post-flight, cultured with normal and altered bacteria, and then we're going to look at a whole bunch of outputs from these cultures, microscopy, cytometry, inflammatory proteins, a whole bunch of things. We're going to be able to characterize how well the astronaut's immune system fights the normal bacteria and the altered bacteria before, during, and after space flight. The eventual goal for all of this work is to lead to a countermeasure for this. We want to keep the astronaut healthy and safe in deep space. And so there is a large international team that is working on a countermeasure to rectify the immune problem during deep space missions, exercise, diet, immunology, microbiology specialists, and those countermeasures will be tested soon in an Antarctica base and then next on the ISS. But for right now, we characterize the infectious disease risk by combining host, which is the astronaut cells, with pathogen, which is the bacteria, and we'll see what happens. I'll stop there, and thank you very much, and I'm happy to entertain any questions.
Thank you. We will now take questions for Dr. Kirshen. Again, please press *1 to be entered into the queue. Dr. Kirshen, I have a question. How has this research built on any previous human research conducted on the ISS?
Oh, that's a great question, and I've been with NASA for about 20 years, and it's taken about those 20 to I've been able to observe all of the various immune and virology and microbiology studies that have characterized this, going back from the shuttle when we were really doing sortie missions. They were very short duration, and we weren't too worried about things like this. With the wonderful advent of the ISS, where we were able to get people in space for 6 months, get them out of that early adaptation phase into what we call space normal, and really see these physiological changes, characterize them, and then start to get the countermeasures. I think it's really wonderful that we're really using the ISS to set the stage for getting people to deep space safely. So there's a whole variety of immune and virology studies. Dr. Clarence Sams, Dr. Dwayne Pearson, our Russian friends, JAXA, Canadian friends, our ESA friends, all have been working on this. It's really a nice international community of immunologists that work to solve this problem together for space.
Thank you. Next, we'll hear from Alicia Carey, Lead Program Manager, Turbine Superalloy Casting Module. Ms. Carey has worked for the Redwire Space for 3 years, focusing on developing commercial in-space manufacturing opportunities for low Earth orbit. Alicia?
All right, good afternoon. I'm really excited to talk about the SDN payload, like she said, Turbine Superalloy Casting Module. It's a commercial in-space manufacturing device that's designed to prove proof of principle for polycrystal superalloy part manufacturing in microgravity for terrestrial use. Superalloys thermally processed in microgravity could have an improved microstructure and mechanical properties over superalloys processed on Earth. So this work expands the utilization of the ISS into a commercial product area not previously investigated. SCM will focus its investigation on potential improvements in superalloy microstructure by heat treating in the microgravity environment. And I know that was a whole lot of detail, so I'm just going to summarize for you that the overall goal of SCM is to leverage the microgravity environment to create improved metal materials for Earth application. Previous research that the Redwire team has done indicates that removing the gravitational effects creates a more homogeneous microstructure, which would improve the material properties of that metal. So that's TSCM in a nutshell. It's part of a few other material science experiments, though. the Redwire team is really proud to launch the first of many SBM payloads to the ISS. SBM is a part of a series of material science investigations called the triplets. They create or process materials in the microgravity environment to produce improved microstructure and remove the effects of sedimentation, which is often seen on Earth. The Ceramic Manufacturing Machine, or CMM, was launched in 2020 and then again in 2021. It was the first stereolithography, or SLA printer, on the space station, and it was the first ceramic material created in space. The other triplet is the Industrial Crystallization Facility, or ICF. It was launched earlier this year, February 2021, as the first large-scale inorganic crystal grown in space. And both of these experiments produced really promising and valuable scientific data about those materials and the process of how to create them in a really challenging space environment. We're focused on commercialization because we want to pioneer the advancement of on-orbit manufacturing and advanced materials manufacturing technology. We believe these are the critical building blocks to space commercialization and the future of private space industry. The triplets will follow a multi-launch technology roadmap to continuously advance Redwire's understanding of the product creation in space. The next golden age of space is built on a strong LEO economy. It's critical for technology development that will improve our life on Earth and enable us to venture further into space than ever before. So circling back, SBM, this is its first launch. It's focused on processing in microgravity for use on Earth. And we're really, really excited to continue the investigation of how to improve materials for Earth in the space environment. Thank you, Monica.
Thank you. Please press *1 to ask a question to Ms. Carey. Ms. Carey, I have a question. Curious, you mentioned commercialization. I'm curious, how does this investigation contribute to the ongoing commercialization of space? If you could elaborate a little bit on that.
Yeah, absolutely. Right now we're partnered with NASA, but we also have some interested commercial partners after we get the data back from this experiment. So ideally, NASA gives us our first start to understand how great of a microstructure advancement can be created in space, and then we continue to work with commercial partners after that point to focus in on creating a certain product. So NASA has an amazing commercialization program out of Johnson that is really helping us with this launch, but the idea is just to understand the science before you apply it to a specific product. So that's the turbine and the TSCM of our acronym. Ideally, we would focus on turbine creation, essentially for the aerospace and power industry. If you create a stronger material, then you can create a material that can be used in high-pressure jet engines. It could reduce the maintenance costs. It could reduce the break of the material, and it also would translate into higher acceptable blade speeds, which would make the engine more efficient and potentially faster. So we need to understand exactly what percentage of improvement that we're going to get in this experiment, and then we would transition to applying that to a specific part. So commercialization is the main focus of this investigation, and understanding what a difference a lack of sedimentation would create and a microstructure change would create in this specific material.
Thank you. We'll now hear from Dr. Imara Ferreira and Dave Reed, Operations Director for the MVP Plant 01 investigation. Dr. Ferreira is a research professor at North Carolina State University. She is a plant biologist by training with several years of experience conducting spaceflight and gravitational biology investigations. Mr. Reed is an engineer at Redwire Space Corporation. He conceptualized the Phytofuse hardware for plant research and guided its development. We'll start with Dr. Ferreira and then go on to Mr. Reed.
Hello, thanks, Monica. So this will be the first plant experiment to be carried out in the multivariable G platform, or MVP. And the unique feature of the MVP hardware, in particular for our experiment, is the ability to conduct a 1G control on the International Space Station simultaneously in the same experimental hardware. So what this allows us to do is to minimize other space-related variables and to focus specifically on the effect of microgravity. For this experiment, we are using the model plant Arabidopsis, which will be grown from seed for a period of 10 days. During this time, the plants will be imaged, so we will have photographs of the plants growing over this period. And then at the end of the 10 days, the plants will be harvested and saved and returned to Earth. Once the samples are returned to us, we plan to carry out 3 different types of large-scale analyses, and this will be to look at gene expression, to look at the regulation of gene expression, and also the protein products. Our goal and our anticipation is that these data sets will provide a comprehensive overview of how plants respond and adapt to microgravity as well as the space environment. And of course, being able to grow plants in space will be essential for long-duration space travel. And now Dave Reed will describe the MVP and Phytofuse in a bit more detail.
Thank you, Dr. Pereira. The Phytofuse Module is one of several different experiment modules developed by Redwire for use in conjunction with their multi-use variable G facility, or the MVP. The term phytofuse comes from a concatenation of phyto, meaning plant, and fuse, from centrifuge. Phytofuse modules were developed by Redwire engineers to enable early-stage seedling growth studies in a variable gravity environment just like yours. Each module houses 3 custom Petri dishes that include a syringe system so astronauts can hydrate the seeds. Each module also includes lights because growing seedlings need light, a camera to monitor growth, and atmospheric sensors. The larger MVP facility itself controls temperature, relative humidity, carbon dioxide concentration, and removes potentially harmful airborne contaminants. Operationally, astronauts inject a nutrient solution into each Petri dish, removes the syringes, then load the Petri dishes in each Phytofuse module. Modules are installed in MVP's twin centrifuge rotors. One rotor remains stationary and one spins at Earth gravity to provide that true onboard control that Dr. Pereira talked about. Plants will grow for 10 to 12 days before the Petri dishes are removed and frozen for return and post-flight analysis. Here at Redwire, we are proud to be supporting this exciting investigation, which promises to yield new discoveries for the benefit of life on Earth. Much of our work on ISS is about exploring how microgravity can positively impact industries, people, and systems back on Earth, and this investigation supports this mission. Back to you, Monica.
Thank you. We will now take questions about the MVP plant investigation. Please press *1 to ask a question. I have a question for either Dr. Pereira or Mr. Reed. Why did you use this particular plant to conduct your research?
I'll take that. So Arabidopsis is considered a model plant, so it serves as a reference for other plants that we might want to grow in the future. And several advantages are that it has a small size, it grows very quickly. We can grow it in many different formats, in petri dishes or in small pots. It was the first plant to have its genome sequenced, and there are many molecular resources and tools for Arabidopsis, which makes it useful for these kinds of studies. Thank you.
Next, we'll hear from Nico Hansen and Teo Nelson, representatives from 2 Student Payload Opportunity with Citizen Science Payloads, or SPOT. We'll start with Mr. Hansen, who graduated with BS in Chemical Engineering from the University of Idaho in 2021 and is currently pursuing a PhD at the University of California, Davis. Nico, can you tell us about your team's investigation?
Yes, so thank you, Monica. So first off, good morning or good afternoon to anyone that's not on the West Coast. I wanted to thank everyone for being here. So first off, as our title suggests, our group is investigating the performance of bacteria-resistant polymers in microgravity. But I wanted to first take a step back and talk about why we chose this project and why we think it's so important. Now, typically when we think about space travel, we think of things like rockets or spacesuits, maybe even lasers. But we rarely think about microbial life and the potential dangers that they pose to long-term spaceflight. Now, the most obvious danger is that of disease transmission, because a crew can spend extended periods of time in close quarters. The less obvious danger is that of biofouling, which can threaten the integrity of certain materials on board. If we think about disease transmission and the spread of bacteria, some of the first things that come to mind are contact with door handles, handrails, and even handshakes. Now, our thoughts coming into this study were as follows. Could the application of bacteria-resistant polymers to high contact surfaces limit the adhesion and growth of bacteria in space? Now, to do this, we selected one zwitterionic polymer and two polyantholite polymers for our consideration. Now, all three of these polymer chemistries have been shown to resist bacteria adhesion and biofilm formation in Earth-based studies. One of the first things that we did was we conducted a citizen science experiment in which we recruited over 200 local grade school kids to run experiments for us. Now, each student was provided with a citizen science kit, which included samples of each of the polymer candidates, as well as an agar control. Students were instructed to rub various household and school surfaces using a cotton swab and apply the residues to the trial samples. Each student then tracked bacteria growth for a period of about 30 days and provided us with valuable data as to the side-to-side performance of each polymer chemistry candidate. We then used this data to narrow our focus to the best polymers available and designed our experiment accordingly. The experiment that we'll be conducting is also partially inspired by the ISS itself, specifically the handles astronauts use to maneuver themselves around in microgravity. The same aluminum alloy that the handles are made of will be used as the substrate that our polymers will be applied to. In this experiment, we'll be assessing the adhesion of the bacteria strain Staphylococcus epidermidis onto both coated and uncoated aluminum samples. I would like to note that Staphylococcus epidermidis is a bacteria strain that was selected due to its presence on human skin and is therefore representative of bacteria transmission via skin contact. The results of this experiment will be compared to a control study that we will run in parallel here on Earth. Now, what we hope to see from the results of this experiment is both a clear reduction of bacteria adhesion on the coated samples versus the uncoated samples, and also that the polymer performance in microgravity does not diminish as it compares to our Earth-based control. With that, I'll turn it back to you, Monica.
Thanks, Nico. Could you tell us what were some of the biggest challenges that your team faced in developing your experiment, and how did you overcome them?
So thank you. I think that the biggest challenge that we had was coming into this with limited experience on how to conduct research in microgravity, which includes testing different mechanics of the experiment and testing it in such a way that we're not relying on gravity, and we're testing it in a way that we can see whether different functions of the experiment will work when introduced to a microgravity environment. Thank you. Turn it back to you, Monica.
Thank you. And as a reminder, for any of the reporters on the line, please press *1 at any time to be entered into the queue. And we'll now hear from Teo Nelson, who is a current student at Columbia College on a pre-med track, planning to major in computer science. Mr. Nelson, can you please tell us about your team's investigation?
Thank you, Monica. I'm very proud to be speaking today on behalf of the Columbia SPOX team. We are a team of undergraduates from all across the world united by our commitment to this project and the future of long-term spaceflight. NASA announced this competition to celebrate 20 years of research on the International Space Station. To have even hope of joining this tradition of discovery and innovation was truly inspiring. We put no limitations on who could join us and work on this project. Students from all backgrounds, disciplines, and ages have contributed to the experiment that you see today. We continue to put no limitations on who can be a part of our project. We promise to make all of our data and experiments available for citizen scientists to analyze. We hope to inspire students and scholars of all ages to join the research community at the International Space Station. Our experiment in particular focuses on 2 of NASA's 5 medically important microorganisms, Staphylococcus aureus and Pseudomonas aeruginosa. The novelty of our experiment is the rigorous study of a dual species biofilm between these 2 bacteria. On Earth, the combined bacteria often cause more severe disease and display more resistance to antibiotics. We believe that this effect will be amplified in the microgravity environment of the International Space Station and therefore threaten crew on long-term spaceflight. Under severe mechanical and electrical constraints, our engineers have designed a payload within a 10 centimeter by 10 centimeter by 15 centimeter enclosure capable of autonomously conducting 3 experiments simultaneously. The first will inject a fluid into specially designed Petri dishes, which will preserve the DNA and RNA of these bacteria before they land on Earth, contributing valuable sequencing data to a field with less than 5 pre-existing comparable data sets across all bacteria species. The second experiment generates live bacteria, which we can use back on Earth for antibiotic resistance testing and assays measuring the response of human cells to these spaceflight bacteria. The third will take the first ever time-lapse of a bacterial biofilm growing in space. All of these experiments will improve our response to this significant threat to space exploration. In closing, I'd like to thank our entire team, but especially my team co-leads, Kalpana Ganeshan and Swati Ravi, for their initiative and resolve at every step along the way. Our advisors, Professor Lars Dietrich and Professor Mike Massimino, along with Columbia University, NanoRacks, and the NASA STEM on Station program for all their support. I look forward to your questions.
Thank you. We will now take questions about the Columbia student investigation. Please press *1 to get in queue. Mr. Nelson, what significant contributions does your investigation make to the field of microbiology as situated within the microgravity environment?
We are studying microbes that have caused us problems for a very long time. In 1970, in fact, Fred Hayes, part of the Apollo 13 crew, developed a severe urinary tract infection which was caused by the very bacteria, Pseudomonas aeruginosa, that we are studying. In terms of our contributions to this field, we've developed a fluid injection system that is optimized to operate in microgravity and preserves a fully sterile and sealed environment, preventing contamination within our controlled experiment. We are also providing significant sequencing data based on the framework of the NASA Gene Lab, which has pioneered many of these standards and investigative technologies. And lastly, of course, is our novel video, hopefully time-lapse video of the bacterial biofilm growing in space, which will provide significant information regarding the increased, or as we hypothesize, increased structural integrity of these bacteria as they are growing together.
Thank you. We'll now hear from Dr. Paul Reichert of Merck Research Labs, who is the principal investigator for the CASIS PCG-20 experiment. Dr. Reichert, can you tell us more about this investigation and how it may lead to better cancer drug delivery?
Good morning, good afternoon, and good evening, wherever you are in the globe. I'll try to give you highlights of our planned SpaceX-24 monoclonal antibody crystallization experiment for drug delivery. Although monoclonal antibody therapeutics have been widely applied to unmet medical needs in cancer, in immunological, and antiviral disease like COVID, there is a challenge to deliver them to patients since they require administration over several hours as IV infusion in hospital settings. That's because liquid antibody formulations have limited solubility and must be given at high doses. To improve the delivery of monoclonal antibodies, Merck and other pharmaceutical companies are investigating the development of injectable formulations rather than the need for IV infusions, which can be administered in doctor's office, like vaccine injections. This would improve the quality of life for both the patients and caregivers at reduced cost. The challenge is to develop high concentration formulations in pre-filled syringes suitable for injection. High concentrated crystalline suspensions are being investigated as potential injectable formulation candidates. Merck, in collaboration with CASIS and NASA, is performing crystallization experiments on board the ISS to identify what are the key variables for crystal growth as well as the preferred properties for an injectable formulation. The planned experiment builds off a published SpaceX-10 experiment performed in 2018 with pembrolizumab, the active ingredient in Keytruda, Merck's immuno-oncology cancer drug, where space-grown crystalline suspensions were more uniform, less viscous, with improved injectability properties than the comparable ground-based experiment. This aided the development of crystalline suspension processes on Earth. The results of this experiment was reported in a 2020 article in Nature Microgravity. The planned SpaceX-24 experiment is further investigating the key variables for crystal growth, such as purity, diffusion, and temperature in custom 3D printed hardware. The hardware was designed based on the pioneering demonstration of liquid handling capabilities by astronaut Dr. Kate Rubins during her 2016 ISS tour. The experiment also involves astronaut activation and observation during the experiment. The ability to recover our experiment under optimum refrigeration conditions in hours from the returning Dragon in Florida versus days when the Dragon returned to Earth from Florida on previous missions. Upon return, we plan on performing a multitude of biophysical and physical characterization methods for evaluation of both the flight and ground experiments. The experimental design of this experiment was meant to challenge and inform other investigators to consider microgravity research as a tool to further their Earth research. Thank you for your attention. Back to you, Monica.
Thank you. We will now take questions for Dr. Reichert. Please press star 1 to be entered into the queue. If there are no questions, we will move forward. And finally, we will talk with Dr. Mike Civic, Senior Director and Research Fellow at Procter & Gamble, working in the Strategic Innovation and Technology Division for Fabric and Home Care. He is the Principal Investigator for the PG Tide experiment. Dr. Civic?
Thank you, Monica. We are delighted to have the opportunity to partner with the National Lab, NASA, and Tide to develop laundry systems for deep space missions for the future. As you may know, astronauts do not do laundry in space today. And as the development of space missions become a more foreseeable opportunity for future life, we need to develop laundry systems that are making more viable. And in doing so, mission PG Tide is broken into two parts to do that. We've been working with NASA over the last 2 years to develop detergent Tide Infinity that meets the requirements provided by NASA to be able to be fully degradable, compatible with closed-loop systems, and obviously be separated and allow water reuse, which is one of our primary objectives to sustain laundry systems for deep space missions. As a part of this study that's going to be launched next week, we want to understand the stability of our Tide Infinity detergent and its components to exposure to microgravity, radiation, and space travel in general. We're running controlled experiments in parallel, and when we receive the payload next summer, we will obviously assess the performance and ingredient activity of its components to determine what is sustainable for flight for longer duration experiments. And this experiment with the National Space Station National Lab allows us to do some significant learning in that regard. Back to you, Monica.
Thank you, Dr. Civic. If you have a question for Dr. Civic, please press *1 on your phone. I have a question. Dr. Civic, how do you see this possibly transforming the future of space exploration and supporting deep space goals?
Well, it allows, well, first and foremost, we're trying to reduce payload up mass for NASA and other space missions. For an average travel to Mars, for example, a 3-year mission would require about 500 pounds of clothing, which is just not possible. So this study will make it viable to reuse laundry today that's not being done at the current moment on the space station, which is needed. We also plan to use learnings from these missions to reapply them to on-Earth needs and requirements. As you know, there's water conservation that's needed around the globe. We're trying to utilize resources more effectively. Tide is a mission for doing just that with our Ambition 2030. We're trying to get consumers to do laundry in much colder wash conditions, and as you can imagine, saving energy and resources is key not only for space travel, but also here on Earth, and we're using this opportunity to rethink laundry. And the constraints put forth by NASA and space travel allows us to do just that.
Thank you. We will now open it up to take any additional questions for any of our speakers. Please press *1 to get into the queue if you have a question. Dr. Reichert, I have a question regarding your investigation. Could you please further explain how your investigation might help people who are receiving cancer treatment and how and where they would receive those treatments?
Our hope would be that we could give the injections in a doctor's office. Right now, a number of patients and caregivers that are going for their infusions are in either hospitals or outpatient facilities where both the patient and usually a caregiver is taking a day off from work and time in order to get these infusions. And I always notice that when I talk about this to an audience, the people that are not really listening, when they hear about the effect that these infusions have on caregivers, I see the immediate response about what it would be like if they could just simply go to their physician and get an injection that would take 5 minutes and would reduce the cost of delivering that infusion, that injection, by 50%. So that's part of what drives me to continue to investigate an opportunity for a single high concentrated crystalline suspension that would also be room temperature stable.
Thank you. And I have actually a follow-on question for your investigation. I'm curious, could you go into more detail on how the return to Florida versus a return to California helps your research?
I'm based in New Jersey, and I've been on 3 previous SpaceX flights, and it was always an arduous task to fly from New Jersey out to California, because that's where the Dragon would parachute down about 200 miles off the coast of California. And I would have to be waiting there usually a few days before the capsule would be picked up by a vessel that looks like a tugboat, and it would take 3 to 5 days to get the vessel back to the dock in Long Beach. It was such a relief to be on this mission and to have, the Dragon return off the coast of Florida and be picked up and have the ability to get the experiment back in a matter of hours is just incredible to me.
Thank you. And thank you to everyone on the line for your interest in this important science, research, and technology that are launching aboard the SpaceX CRS-24 mission next week. You can follow all the research being conducted during the Expedition 66 6-month mission by visiting nasa.gov/station research news. That's nasa.gov/station research news. Or follow ISS_research on Twitter. Please stay tuned to nasa.gov/commercial crew for regular updates in advance of the upcoming launch. We are targeting Monday, December 20th at noon Eastern Time for our pre-launch news conference broadcast. Launch broadcast coverage will begin at 4:45 a.m. Eastern Standard Time on Tuesday, December 21st. As a reminder, you can listen to a replay of this teleconference by dialing 866-360-7721. That's 866-360-7721. Or by visiting the NASA video channel on YouTube. Thank you all for joining us.
This concludes today's conference. Thank you for your participation. You may disconnect at this time. Speakers, please stand by.